In this video, we are going to discuss protein's structure or the shape of the protein under AP Unit 1 Chemistry of Life. In the previous video, we said that from 20 amino acids, we can have a wide array of proteins. And we can better understand it by relating it to the 26 letters in the English alphabets. From just 26 letters in the English alphabets, we can create millions of words. So similarly, from the 20 amino acids, we can have hundreds of thousands of proteins. But proteins may have up to 4 levels of structural organization. So we can talk about primary protein, secondary protein, tertiary protein, and quaternary protein. Let's start discussing the primary protein. So primary protein is simply a linear sequence of amino acids joined by peptide bonds. However, this sequence of amino acid or this polypeptide can fold to form a secondary structure due to the hydrogen bonds that are possible to be created after every 4 amino acids. So after every 4 amino acid, it's possible to create a hydrogen bond thanks for the polar peptide bond, polar covalent peptide bond that allows the amino acids to create hydrogen bonds. And due to these hydrogen bonds, the primary linear structure will form an alpha helix spiral, as you can see it here. So it forms an alpha helix structure, and again the hydrogen bond that occurs at the 4th amino acid gives the protein the possibility to form this kind of alpha helical structure. But in the secondary structure, it's not just the alpha helix. We can also see something called the beta sheets, where the protein turns back upon itself and hydrogen bond will be formed between extended lengths of the protein. And it's necessary to mention that in secondary structure, we can talk about fibrous proteins. Fibrous proteins can exist in alpha helical structure and in pleated sheets as well, such as the keratin and silk. So if you are going to address the fibrous protein, usually the fibrous protein have the secondary structure in both forms, the alpha helical and the beta sheets. Now these secondary structures can fold more to form a 3D shape of polypeptide. This 3D shape of polypeptide is called the tertiary structure. So by definition, the tertiary structure is a single polypeptide that folds more into 3D shape to form a globular protein. But what's the reason behind this? Why the protein has to fold more into 3D structure? This is because most of proteins are found in aqueous solution. So you would expect the hydrophobic amino acids to hide into the interior side of the protein, while the hydrophilic amino acid will be oriented towards outside or the surface, and they are in contact with water. So again, the tertiary structure of a protein is actually a 3D shape of a single polypeptide. And the reason behind it is the fact that many proteins are found in aqueous solutions or aqueous environments. But what kind of bonds do you expect to have here? Of course, you would expect the hydrogen bond, because as we said before, hydrogen bond is found in the secondary structure, and the secondary structure is part actually of the tertiary structure. You can see the alpha helical structures and the beta sheets. So hydrogen bond is expected to be in the tertiary structure in addition to the covalent bond. Think about the peptide bonds, in addition to ionic bonds, and most importantly, the disulfide bond. So due to this extra folding, or the folding into 3D shape, it's possible for 2 cysteine. Do you remember the cysteine amino acids? They have the sulfide bonds, the sulfide functional group. So it's possible that two cysteine amino acids come near to each other and create a disulfide linkage. So you would expect also to have a disulfide linkage in a tertiary structure, and this kind of bond is necessary to maintain the tertiary structure. One example of tertiary structures are the enzymes, the globular proteins that ball up into 3D shapes. Now, tertiary structures are also found, we can expect to see tertiary structure in quaternary structures. So by definition, the quaternary structure is made up of two or more folded polypeptides. So the major difference between quaternary and tertiary is that tertiary made up of a single polypeptide that fold into 3D shape, while in quaternary you would expect to have two or more folded polypeptides. And even more, you can find up to 4 polypeptides, as in the example of hemoglobin. So quaternary structure is also globular protein that might have 2 or more folded polypeptide. In the case of hemoglobin, you would expect to have 4 polypeptides found there. And again, the bonds that you would expect to see here are the hydrogen bonds, the ionic bonds, the covalent bonds, and the disulfide linkages.